Sam has a background in theoretical quantum physics with a PhD in quantum dynamics and thermalization. He also has done his postdoctoral research in the simulation of open quantum systems. He has spent many years supporting commercial R&D organizations with cutting-edge technologies. So over to you, Sam. You can please proceed with the session.
Thanks very much. Thank you for the kind introduction and also for the invitation to speak at the session. So as you mentioned, I've worked in R&D for a long time, and my background before that is in quantum technologies. So actually, this is a really exciting time for me. It's fantastic to be able to discuss how these areas come together and how we can talk about an area like life sciences R&D and a quantum view on that. So let me begin by just giving a little bit of an overview of the plan for this session. First of all, I'd like to give an introduction to everybody just on some of the challenges in drug discovery and in general the sort of process of life sciences R&D. So a little bit of an introduction to the industry. I know that on the call we've probably got some people with a life sciences background, maybe some with a quantum background. So this is to kind of bring us all up to the same level. Then I'll talk a little bit about quantum computing and drug discovery applications, and I'm going to focus a little bit on what people are looking to do right now in the NISC era, the sorts of things that they are exploring. I'd like then to leave a little bit of time to discuss a couple of the projects that we have in our internal research in Capgemini. And at the end, you just give a couple of slides to leave you with a few thoughts about how I think we fit in into this industry regarding quantum. So that's the plan for this session. So let's kick things off by talking about drug discovery and the challenges. So for those of you who don't know the industry, it's an industry with an over $1 trillion annual revenue. This is not too dissimilar from other organizations, which are other industries which are R&D heavy. But the interesting thing about pharmaceutical R&D is that most of the products actually fail before they reach the consumer or before they reach the patient in the case of drug discovery. So around 90 to 95% of drugs which have made it to the clinical trials phase will not actually progress and become approved therapeutics. Now, this wouldn't be such a problem if it weren't such a challenge to create these drugs in the first place, but it takes around 10 to 12 years or more to undergo that R&D process. The cost is huge. It's over $2 billion. And so there are real challenges in this industry. Now, before we go on to talk about those in a bit more detail, let's just take a bit of a look about some of the players. I think these are all names that you'll know, and you'll notice that they are all multi-billion-dollar organizations, and that's really because to take on the risk of end-to-end drug discovery, you have to be at that scale in order to do this. So while there are smaller biotechs that maybe focus on some of the research but don't take products into clinical trials, there are organizations which license molecules which have already been created by other drug discovery companies. This sort of end-to-end pipeline is a big business. And it's worth pointing out just at this point that pretty much all of the companies on this list, most of them are making some investments in quantum computing today. They're already starting this exploration. So why is it such a challenge? Well, the challenge with drug discovery is that once you bring a product to the market, you only have so many years before the patent on your drug expires. So at some stage during development, when you reach a drug compound or a series of compounds which you're going to progress, these will get patented. At that point, you have usually 20 years before generics companies can start making generic drugs, and of course, this lowers the price for that particular therapeutic. So the rest of the R&D process, the optimization, all of the clinical phases and approval have to be done quickly and in a cost-effective way, so that you can maximize the amount of time that you have to recover those extensive costs in R&D. And this has been a challenge for the last several decades, that actually the number of new drugs actually created per $1 billion R&D spending has been falling exponentially for quite a period of time. So if ever there were an industry that needed a revolution in compute and was sort of seeking new technologies to change the way in which they conduct R&D, life sciences organizations are indeed part of that, and have been. I want to talk a little bit about the drug product life cycle. So I want to just introduce you to the sort of stages of R&D, so that when we talk about applications, you'll get a picture for kind of how some of these fit into the overall picture. So briefly, at a high level, the process starts with identifying biological targets. These are the entities, usually things like human proteins, human RNA, which you are going to try and find a therapeutic which interacts with in some way to alter the course of disease. So this is the process of linking the disease to the biology, so that you can actually start to create a drug. As we move along this pipeline, we start by finding trying to identify leads, and this explores often searching millions, actually now in some cases billions of potential potential compounds, potential molecules, potential biologics. And then we progress. When we identify some which show some activity against the target, there's a big lengthy optimization phase. I'll talk a bit more about that in detail. We then go to preclinical stages, where we talk about safety and actually understanding how therapeutics behave in vivo, and then of course a lengthy clinical phase to assess safety and efficacy, and hopefully after all of that time an approved drug. So we're just going to step through these areas over the next few minutes, just to talk about some of the challenges which are faced, so that we can then get a bit of a picture of what pharmaceutical organizations are really trying to go after in R&D. So let's begin with target identification and validation. This is all about understanding the biological processes involved, and ultimately which things you can actually interfere with, by bringing in a foreign therapeutic into the body to interact with. So the sorts of challenges which are involved here are everything from text mining to identify targets from extensive literature, right through to looking at genomic analysis, which is becoming increasingly relevant as people seek to make genetic links with diseases and use that as a way to identify targets, and particularly with a view of personalized medicine. Reasoning on networks, so identifying biological pathways and trying to identify which the entities are which can be interrupted to have an impact on a disease. And finally, actually understanding which of those are likely to be possible to find a drug against. So it's quite important that to note that not every target is necessarily leads to a successful outcome down the pipeline. It has to be of a certain structure, such that it's possible to actually create therapeutics that will work. Okay, so that's the first stage. From that point, once that's identified, the really large scale broad search for potential candidates begins. This is where there's a huge amount of robotics, a huge amount of screening effort. You can see here these kind of these micro titer wells with robotic pipettes. High throughput screening labs can screen several million compounds in over a period of a few weeks, but it costs a few $1 million to do that. Newer technologies like DNA encoded libraries, where you actually tag molecules with DNA so that you can retrieve them later, can push this to the billions. So a huge amount of data is generated here, and there are lots of challenges in optimizing this approach. And more recently, we've been working to actually complement that with virtual approaches, where we use models which predict the outcomes of these screens, because although a billion sounds like a big number, for small molecule drugs, actually the size of chemical spaces is thought to be around 10 to the 60 potential drugs. So we're really dealing with a drop in the ocean in terms of the amount that we can actually screen, and if we can expand that, then that's incredibly valuable. So you'll see that later when we talk about some of the quantum applications, this certainly forms a part of that. The next phase, another area we'll talk about in quite a bit more detail, is lead optimization, because it also has some quantum applications. This is around taking some of those leads, some of those molecules which have shown some activity against that biological target, and improving them, because it's not enough for them to just bind to a protein or other entity. They need to show a good level of efficacy. They have to be suitably selective, so they They must essentially not bind to all sorts of other things in the body, not be a risk for toxicity. It's not just enough to bind to the protein that you're interested in. It also has to be safe and not interfere with other processes. And indeed, all sorts of other things, including sort of solubility, synthesizability, many properties which have to be optimized together. And this is necessarily an iterative process of sort of making changes to molecules, making changes to molecules in a sort of design phase, actually synthesizing them, testing them, looking at the results, and kind of repeating this process. And there's a huge realm of opportunities here, using predictions in silico. Again, this idea that if you can do some of this on a computer, not in a lab, you can make predictions of molecular properties, you can predict their activity. If you can simulate it, for example, use data from previous studies to find relationships between molecular structure and activity. These are all routes to go. And indeed, within this, there is just a big optimization challenge of all the possible things that you could make. Which things should you actually go ahead and make? And we'll revisit that in a little bit. So next is preclinical, and this is really about assessing safety. It's about animal models. It's about modeling how that drug behaves in the body. How long does it stay in the body? How quickly is it metabolized and excreted? How often do you have to dose a patient in order to keep the concentration of that at a suitable therapeutic level? Obviously, a product isn't much use if you have to dose somebody sort of 8, 10, 12 times a day. All of this kind of dynamical processes in the body are important, as is safety. So looking at sort of pathology, looking at these sorts of modeling activities are hugely important here. And when that's passed, finally, it goes into people. And we start to understand, firstly, the safety, then understanding the sort of dosing in phase 2 clinical trials, and finally really getting a good picture of the efficacy of a drug at scale in a large population is really the sort of the sort of final stage of a phase 3 clinical trial, where you'll also be looking to check that there are no detectable rare adverse events as well in that bigger population. And there's a ton of optimization work to be done in this phase. So everything from the trial design, so that you can get data as quickly as possible, that actually helps you make a decision about the drug, helps prove that it's effective. Also optimizing the trial for cost, for patient burden, minimizing the number of times you have to do a blood draw on a patient, for example, in order to get the information you need. Lots of optimization there. Also at the operational level, which sites to use to get the right patient populations, how are you going to optimize the supply chain, which patients are going to benefit from a particular therapeutic, coming back to that personalization. So tons of optimization in this area. And finally, even though it's not necessarily considered part of the R&D, and I'm not going to come back to this really in the applications area today, manufacturing, going from small pilot scales, scaling up from the lab bench to manufacturing, is something where we also think actually there are some certainly longer term potential for quantum computing. If you look a little bit further into the distance, there's a lot of challenges around this space as well. So all of that was a quick whirlwind tour through the R&D process at a high level for drug discovery. This had a slight sort of bias towards small molecule drugs, but more or less the same kind of processes apply also to biologics as well. So with that in mind, let's move to talking about quantum computing and some of the drug discovery applications. And just as I apologized at the start from anyone from the life sciences background for the first overview here, I'm going to have to make an apology to everybody who's involved in quantum computing, that I'm just going to give a basic introduction to how quantum computers are different. I know many of you on this call are going to be completely familiar with that, so feel free to zone out for a couple of slides, and then we'll pick things back up again. So quantum computers and classical computers, I think we all know that the classical computers, the sort of conventional devices in front of us, in our phones, in big HPCs, they deal in bits, ones and zeros. We perform operations on those. We can do everything from managing databases right through to exotic bits of AI using this technology. Face value, quantum computers look similar. Qubits instead of bits, quantum bits, zeros, ones. Those funny brackets around them signify that these are quantum states. But where the differences start are that these can be in other states as well. They don't just have to be 0 or 1. They can be in things that look like 0 plus 1 or 0 minus 1, or even with imaginary amplitudes here. So there's a whole host of potential superpositions. And for those of you who are coming from outside of the quantum world, this is exactly what people are talking about when they talk about Schrodinger's cat, that this is exactly the phenomenon of superposition. Now, at this level, it doesn't seem particularly interesting, but if you start to move to a larger number of bits and think about the quantum equivalent, if we go to 20 qubits, then a general state of a quantum computer can be a linear combination of with all of these different amplitudes, A1, A2, A3, of all of the different combinations of those ones and zeros. And actually for 20 qubits, we have 2 to the 20 of those. And I think you can get where this is going. If we go to 40, we're going to have 2 to the 40, which will take us from 1,000,000 to 1,000,000,000 amplitudes. And sure enough, if we think about maybe a few hundred qubits, then actually the number of amplitudes there in general is more than the number of particles in the universe. So we know we can't even represent it ever on a classical conventional computer. And the challenge of quantum computing is using the operations that we can do on quantum computers to make some use of this, to actually be able to do clever things with this superposition of bit strings, which entangle those ones and zeros together, allow for superpositions, do clever things with the with these essentially in such a way that we can get useful compute out the other side. So if we take a bit of a step back again to a sort of high level again, the sorts of challenges that life sciences R&D organizations are exploring are the same as other organizations. They're looking at simulation, they're looking at optimization problems, interesting combinatorial problems, and they're beginning to look at where quantum machine learning might have an impact as well. So where are we now with this? I'm just going to take a few slides to talk about where we are in the current NISQ era, and then talk a little bit about some of the things which people are exploring in the industry already. So the NISQ era, for those who don't know, is the noisy intermediate scale quantum era. It's where we are today, with qubits numbering to around 100 or so. In a couple of years, this will be more like the 1,000 or so. But we don't have enough qubits to do massive scale computations, and we also don't have perfect qubits, and we're unable to correct their errors as they go. So when we prepare qubits in a particular state, when we perform operations on them, in fact, even when we just leave them and don't do anything, they begin to decohere. All of these things are not perfect, and they mean that we can't we can't do endless calculations with them. So it's a little bit like if you imagine having a pocket calculator and you're typing into it and you keep going, and then eventually the screen starts to scramble and you figure, okay, I need to find a way of writing shorter sums. And that's what a lot of the effort at the moment is all about. So the next 2 to 3 slides will be very slightly more technical, but then I promise we'll come back to the high level, and they're not going to be super technical at all. So NISQ era quantum computing is all about being able to create a series of operations on qubits. So these 4 lines across show the things that we can do to qubits. We can change their states in particular ways. And there's a limit to how many of those operations we can do before we actually I either see our qubits decohere, which means that we lose a state in them, or we get a buildup of errors from all of these particular operations. But at the end of it, we take a measurement. And the nice thing is that although when we take a measurement, we either get a 1 or a 0, we get a collapse. Even in this NISQ era, we can do quite a lot of these. So although we can't do circuits which are very deep, we can repeat something lots and lots of times and do multiple shots, which is potentially helpful. So for those who are interested and know the sort of background, essentially what we're doing, all of these operations are parameterized unitary evolution of the qubits, and then we can project on each of those. And I thought it'd be interesting to just consider the sort of general class of problems which people are exploring how to solve in this NISQ era, because quite a few pharma organizations are starting to look at the kinds of challenges and just play with them at toy scales. It turns out the sort of relevant problems you can do involve effectively finding low energy states of a system, which is a very abstract thing to say, but that can mean, broadly speaking, finding the state of all of the electrons in a molecule like this. So doing quantum chemistry. It may also be possible to map it onto a problem in combinatorial optimization, where here's a classic knapsack problem. You are looking at trying to maximize the value of the items that you can carry in that rucksack when they've all got different weights. And it turns out that there are definitely some differences between these, but they both are look a little bit like finding low energy eigenvalues of an effective emission or symmetric matrix. Now, if these things on the bottom don't mean anything, don't worry about it. But the parallel I'm just making here is that sort of the best configuration of this is the lowest energy eigenvector, and to some extent also the state of all of the many electrons in a molecule is also such a thing. So I just want to sketch a little bit how you do this using an algorithm called VQE, the variational quantum eigensolver. Many of you will know this, but it's just, I think it's worth mentioning because a lot of people are studying this for some of these near-term problems. And I just want to sketch out at a high level what you're actually doing, because it's an algorithm which leverages both quantum computers and classical computers. So the first step of what you do is you take your business problem and you map it to qubits. And don't worry about the maths here, but this is essentially a general form where you'll have different values of alpha for a particular real problem here. Whole host of problems can be mapped to qubits. You'll find some way of identifying a whole load of parameters which you can change with a classical computer. So a couple of slides ago, I showed a series of operations being applied to qubits before we make measurements. We need to find a way in which we can actually tune those and tweak them. And then when we have that, we start to actually do some things on a quantum computer for real. So we enact a series of gates which prepare a quantum state, and then we make a measurement. And we can do that lots and lots of times, and actually doing that gives us a way to gauge the energy of the system for that particular state. We'd like to get to those low energy states because those are the problems, those are the ones which provide the solution to our problem, whether it's the configurations in an optimization problem or whether it's the electronic structure of a molecule. And this is something we can do classically. We can look at all of the parameters that we used the first time and say, which would be better parameters to try and get a little bit closer to that ground state. And then this is a cyclical process where we keep going round and round this loop. And that's really at a high level kind of exactly what we're doing. And the bit, it's worth noting, I guess, the bit that we're doing on a classical computer here is the sort of thing which we are used to doing on classical computers when we optimize things like neural networks. If we squint, they're sort of similar kinds of optimization problems at a high level. Okay, so that's the sort of slightly deeper dive into the kinds of algorithms that are being looked at the moment. Let's come up back a level and talk about what people have actually been doing. Let's first of all talk about simulation. People have been studying simulation at small scales. This goes back to 2017 from IBM, where they looked at small molecules, things like hydrogen, beryllium hydride. Now, it's worth noting these are not drug molecules. These are significantly smaller than drug molecules, but they sort of demonstrate the principle. More recently, Roche, in collaboration with Cambridge Quantum, have been using essentially these kinds of approaches to get the first possible step on the ladder, if you like, to use a quantum computational approach to study interactions of drug molecules with proteins. So they want to look at the binding energy, the difference between the energy of a drug molecule when it's near a protein versus when it's not near a protein, because ideally, if a drug is going to be effective, it should bind strongly, and that's something they're trying to predict here. So they took a series of molecules that they already understood. Now, scale of quantum computing is not anywhere near the scale where we can study a whole molecule, but instead what they did was they took a fragment-based approach. And actually this blue region here, this small region here, which is the bit which binds to the protein, they studied on a quantum computer, and the rest of it was actually studied in an approximate fashion. Now, we'll come back to this topic of simulation because there is really quite an interesting question as to how far you can push these NISQ-era algorithms. But nonetheless, Roche were able to show that they could correctly, more or less, rank the molecules in terms of their potency by simulating and finding the energy of binding with the protein. So on the y-axis here, you've got this sort of output from the quantum computer, and on the x-axis here, this PIC50 is a measure of how potent a drug is, and they more or less agree. So worth saying, nothing here you can't do classically today because we're talking at very small scales, but it's nice showing the sort of end-to-end picture of how these things fit together. And being able to recover this plot is a really, really nice step in that direction. Another area we think we should cover an optimization example, and the sort of the headline grabbing stuff is often around protein structure. This is an IBM paper from last year where they were able to put a protein on a lattice. So this is a model where you can constrain where the amino acids are. You also actually constrain the amino acids to be just single nodes in a chain. But they showed that they could correctly use optimization algorithms to find the structures of these proteins on using NISQ algorithms on actual quantum computers. Now, for me, I want to make this clear. I think it's really nice that they're exploring this, and I think it's a fantastic opportunity. But it's worth saying there are a lot of approximations in this in terms of the model, the fact that they're using a lattice model. 10 amino acids is not very many today, and actually I don't think personally that protein structure is probably a near-term goal for quantum computing, but nonetheless it is nice to sort of show some vision and sort of begin to be sort of piecing these things together. So I think it's definitely a case where it's important not to fall into the trap of overhyping the commercial value of some of the research at the moment, however interesting it currently is. Now, some of the other areas for optimization, I was just going to pick out another one in detail that I've seen a few organizations mention. I think is quite interesting to study because it's a problem I've encountered many times in the life sciences domain, is that of building interpretable models. The idea that if you want to create a diagnostic model or even just a prognostic model predicting what is going to happen to a patient in the months and years ahead based on what you measure when they first walk into the clinic. Now, that's incredibly important for personalized medicine, for getting the right treatment into the right patient. There's so much clinical data available. You have tons and tons of measured quantities, which you can use with machine learning to make a prediction. The challenge is that rapidly these models become such that physicians, the doctor you talk to in the clinic is not really able to understand what's going on. So an interesting objective is to try and find an optimal set of biomarkers, which is small, but highly effective. So each of them is relevant to predicting the outcome from disease or predicting the outcome from treatment. But there's very little redundancy between each of them. So we can use various measures to explore this. It's a combinatorial problem that grows very, very quickly with the amount of, the number of different clinical measures we have. But the aim is really to find a set where, there's very little overlap between them. So the sort of bright blue squares here, the darker blue squares here show that there's shared information between each of these variables in this clinical data set. And you can see here where if we were to pick an example where they're pretty light, that would be good because they don't share much information. Whereas over here, they share a lot of information with each other. So there's a lot of redundancy and that wouldn't be a good biomarker panel. And of course, I can jumble up all these rows and columns and there's a huge number of combinations here. So this is one of the applications people have been exploring in this kind of NISQ era for optimization. It's worth pointing out these are heuristic algorithms. There's a lot of debate about how these are going to scale and for different problems, whether or not they. The good news is quantum computing is developing very rapidly, so we're going to be able to see more of that over the next few years. Anyway, now I want to allow a bit of time in the sort of last part of the talk to talk about some of the things that we've been looking at and a bit on where we fit in. So I just thought I'd give a quick overview of some of the other things which I think are interesting. We've seen from GSK some work on codon sequence optimization. This is really about creating mRNA sequences which maximize protein expression. So another optimization challenge. When you go to produce biologics, you want to have really high yields in your bioreactors. This is one of the things to think about there. They've also looked at mRNA folding prediction using optimization techniques in the NISQ era. I haven't put particular papers, but things like site selection and supply chain, these are the kinds of problems that people talk about quite a lot, so they get quite a lot of attention. I don't think I probably need to give them any more attention than they already receive, but a few other interesting areas are people exploring QML, so generative models for lead optimization, something we've worked on a lot in the AI space. but actually how quantum might be able to enhance those. And again, areas like medical imaging. And although in the quantum lab we've not, I think, applied these techniques to medical imaging, on the team we've got people who have applied this a lot. So Barry has looked at QML for imaging a lot, and we have those, we have those capabilities internally. Okay, let's talk about projects in Capgemini. So I want to pick a couple of things that we've looked at. I'm just going to give a little bit of an overview of them. The people involved at some point, I'm sure will be delighted to give a longer talk on them, because obviously in a few slides I can't really do them justice, but it's just to give an overview of what we were looking at, why, and what we found out. So the first one I want to share is some work done by Francis Newson on the team. So I think Francis might be on the call, but we looked at this challenge of essentially whether quantum simulation of small molecules is useful or not. And actually Francis spent quite a lot of time exploring this area. And we came to the conclusion, based on trying to do it for real and also exploring the literature, that actually the scales at which quantum computing is relevant in the NISQ era are a little bit bigger than you might think. So actually there is, of course, if you look at the crossover here, this is a paper from Q&Co, I can share the reference. There is a crossover where quantum computing wins. And so there are some scales where, where it might be possible, might be competitive to do quantum computing for simulation of molecules. But actually the sort of scales for drug molecules are quite a bit higher. They're quite a bit larger than that, and we're talking about quite long time scales on this yellow curve at the moment, sort of today, if you like. And so we asked the question, well, what else can we do in this area to bring it a little bit closer? And so Francis then moved to talk to study the question of quantum machine learning and the advances that could bring, actually specifically quantum kernel methods, to see if we could get some advantage from looking at some of the challenges we'd like to look at, but using those sorts of approaches. So I'm just going to give a one slide sort of primer on some of these kernel methods. These are approaches, they've gained a significant amount of attention in the last year or so. And they're essentially a way of constructing quite flexible models. They're a way of projecting your data into the really high dimensional spaces that you can create with quantum computers, because a few qubits create a space which is sort of exponentially difficult to simulate on a classical computer. And a lot of people have been talking about the sort of acceleration potential there. So a couple of recent studies that motivate this. IBM published a paper that's been talked about quite a bit, where for a very specific problem they demonstrated an advantage using quantum algorithms for machine learning, though this is worth emphasizing is an incredibly specific problem. But lots of other people since have been starting to see, well, what would this mean in our area? And you see examples such as CERN, where a group in high energy physics has looked at detecting events on the Large Hadron Collider and so on. So there's some good reason to explore this and see what it's all about. The specific problem that Francis looked at was actually predicting toxicity. There are standard data sets for this, containing a whole load of drug-like molecules. These include lots of properties associated with them. These are all classical data, things like solubility, surface area, the weight of a molecule, all mostly sort of physical attributes of a molecule that you can use to build predictive models. And Francis actually looked at kernel SVM, quantum kernel SVM. These are support vector machines, because you could do a direct comparison between a classical kernel and a quantum kernel. So what does it look like with the sort of traditional approach versus what does it look like if we project our data into this high dimensional space? And really to explore, what does that look like? Does it look the same as a classical one? Is there a vision for how that might take us further along the route towards quantum advantage in future? So what we actually found was that, yes, at a small scale, the approach is competitive with classical. We would not expect to get particularly better performance. We may, for some specific problems, find that a classical kernel is better than a quantum one because of the nature of the data, but more or less, depending on exactly the data, these sort of two approaches are similar, and that's not a problem because we're looking at data really on sort of toy scales today. And we found that the models are indeed pretty flexible. You can get a wide range of solutions from quantum kernels, and they genuinely look different. So this is just a toy example in 2 dimensions. What you're looking here is a sort of binary classification. So you can imagine sort of blue versus orange being tox versus not tox, but it's a sort of toy example here, and the different colors showing where you predict for a couple of inputs on the X and Y axes, and you can see these are really quite different models. The nice thing I think for us, which is where we want to go next, is that if we wanted to do machine learning on quantum data, well, actually, these are kind of the only kind of approaches we can really use, because I already said that quantum data, once you get to a large enough scale, it's just impossible to actually even store on a classical computer. So if you want to do machine learning on that data, it's going to have to be an approach like this, where the data is already encoded in the qubit. So that's work which is coming next, which Tom Montgomery is going to be leading. And Tom Montgomery also had a role in the second example I'd like to share with you today. Tom continued some work by Scott Ladenheim on optimization for lead optimization. So everybody's studying optimization. We thought we really should map a new problem in the life sciences domain into the quantum optimization space. So we had a look at what hadn't been done and thought, let's study a problem end to end and see what we learn by doing this. And the challenge was around picking which molecules we should go and synthesize in the lab to get the maximum information. So in this lead optimization phase, often you find a scaffold for a molecule, which is a good starting point, and then you have a lot of choices for which groups to go on here and which groups to go on here. And sometimes there can be more than 2 positions where you want to tweak the molecule around and see all the different combinations. But in this data set, there are just 2. This is just a fraction of it. It's actually a data set of 2,500 molecules. And the sort of number you might want to make in the lab is, say, 50. So that doesn't sound a lot, but actually there are already something like 10 to the 27 combinations that you could do that. So 1 and 27 zeros is a big number. We can't deal with problems on that scale, but even on smaller scales, the number of combinations when you start putting one functional group here, one functional group here, and I want to select n of them, it grows quite quickly. So that was our approach, to try and explore this and frame it within that sort of lead optimization process within drug discovery. So just a brief primer on the quantum optimization approach. I've already mentioned the variational quantum eigensolver. This in the NISQ era is one of those problems which people are trying to explore. There is an algorithm called the quantum approximate optimization algorithm, which lends itself to these challenges, and it can be applied to quite a broad range of challenges in combinatorial optimization. And we found that we could map this problem actually in more than one way onto a quantum computer and use this algorithm. It's worth saying that problems that can be tackled with this kind of algorithm can often also be explored with quantum-inspired hardware and with other techniques as well. So that's our aim here, to apply this to this lead optimization approach. How does that really fit in? If we look at the actual challenge, how does it fit in with what we're trying to do in drug discovery? Well, in this lead optimization phase, what we're trying to do is get to a diverse set of chemically potent molecules. Now, it might be obvious that we want a molecule that's really potent because that's going to be a good drug, but actually we want more than that. We'd like to have a few candidate molecules which are different from each other. And that's really a de-risking process, because it means if one of them fails, hopefully the others won't. And if we end up with a few molecules that are all very similar, if one of them fails for a particular reason, whether it's tox, whether it's something else, it probably means they're all going to fail. So that's a big problem. So the aim of this optimization is to get to molecules which are potent, but which are also quite different from each other. And this is an iterative process. We go around a cycle, we run an optimization algorithm to find molecules which are diverse. We synthesize them, we test them. Okay, we didn't really do that. We used a data set where that was already available. But in the real world, the chemists, the medicinal chemists would go away. They'd make those new compounds, probably never been made before, ever. They test them in an assay, and then we get some data. And actually what we can do then is train a predictive model to predict the activities of other molecules. Now, at first, when we've got a small amount of data, that model is not going to be perfect, probably going to be pretty poor, but hopefully it's better than guessing. And it means the next time we run the optimization algorithm, we select molecules which are diverse from each other, but also predicted to be active, to have a high potency. And as we keep going around this cycle, of course, the predictive models get better, and and we hopefully end up with a set of molecules which are both highly potent, but also different from each other. So we did this obviously at smaller scales because the number of qubits available today and the sort of circuit depths are not huge. But we mapped this end to end. We explored which of the various mappings would work effectively. And you can see this is a sort of example of the toy here. This is a sort of combinatorial array of 20 molecules where we're selecting four in the first iteration. These are their potencies, higher is better. In the second round, we select different ones, and we do get a higher potency. And really what we want to be able to show is that, yes, we're able to get greater diversity than you would just by sampling, and indeed that we're able to progress to sort of get better at predicting those more potent molecules as we progress. Okay, in the last part of this this talk, I'd just like to share a few thoughts on where we fit in with a view to the life sciences, given everything I've told you about the sorts of activities which are going on already. So I haven't really taken an industry view. I've talked about some of the specific challenges which organizations are looking at. But let's look at it from the perspective of a quantum journey. Our customers are already in life sciences actually starting on this, starting to assess the potential of quantum technology, finding a few experts in their organizations to start exploring where some of these applications might be in future. And the thing I want to emphasize is that we really are able to help end to end with this journey in Capgemini. And by the time we get to stage 3, we're really talking about getting hands-on with use cases, with algorithms, with trying to explore how they fit in to the business processes, translating from business problems to a quantum computer. And from this point onwards, of course, creating partnerships with the tech providers and scaling up. This is also something we are able to do with our wide ecosystem, both in quantum and also much more broadly as an integrator in areas such as cloud computing. So when I sort of look at us and sort of reflect on how we sort of fit into that journey, I think we really genuinely are a good partner to go on that journey with these organizations. We have a lot of knowledge of life sciences R&D, which is essential. We can speak their language and discuss the challenges on their terms. As this community knows very well, this community is in fact the living proof of our quantum expertise and our really rapidly developing capabilities, and the fact that we're really well placed to help organizations explore, get started quickly, not get locked into vendors, and of course again that ability to scale to really do the end-to-end piece. So I think it's really quite an exciting time for us to be thinking about R&D in these areas with quantum technologies. In terms of the landscape, we do, especially in the sort of target identification and lead optimization phases, think that there's some interesting opportunities here. Essentially, where chemistry is involved, it's a natural place to think about quantum computing. But actually, if you step back further across the pipeline more broadly, there are areas to really delve into in detail. And actually, I think we think that it's not just use cases and sort of finding the use case. It's really about going on that journey of exploration and understanding the technology. Because if we look at similar technologies from a few years back, like AI, the companies that were able to get real value in R&D from that technology were the companies who were able to bring it into their processes, really develop understanding early, so that they could do things which really put them ahead of their competitors using the technology. to what that might look like, this is really just an example, and I'm not going to go into detail on this, but you can imagine a few different themes, whether it's virtual screening and predicting properties of molecules, through to looking at where there's scope for optimization, maybe considering when will simulation be relevant for your business problems, and kind of looking at that. There are a whole load of streams where you can take steps along that journey, and indeed some of the outcomes of these explorations would help feed into all of the others as well. So I think it's now is really the time to start this journey and begin to become ready for the massive scale up of quantum technologies over the years ahead. So I think that brings me nicely to the end. I'll just summarize briefly by saying huge potential in quantum computing in the life sciences. Hopefully what you've seen today has convinced you that there's lots of interesting things to look at. Pharma R&D organizations are actually already starting some level of exploration, and they're probably a little bit ahead of some industries, at least in terms of thinking about quantum. These nearer term algorithms, I do want to give that caution again, and hopefully there's a little bit of a caution in Francis's project when we actually look at some of the scales for quantum simulation in the near terms, that yes, many of these are heuristics. The advantage is that it does have uncertainty associated with it. The good news is that the hardware is developing quite quickly, and so we will be able to progress to explore at bigger scales over the next few years, and that's going to be a really exciting time. And the final thing I just want to leave you with is the idea that we are really well placed to support these organizations in this journey in these exciting times. So with that, I will come to a close and back to the chair.